Hello, and welcome to this webinar where we'll talk about the role of nutrition and brain development. During the course of our webinar today, the key objectives that we'll focus on is to spend some time understanding the importance of nutrition in the structural development of the brain, but also take time to understand what is the role of brain signaling in early life. And that is communication between different parts of the brain, but also how the brain sends messages to the external world and how we take messages back to the brain from the external world. We all know that nutrition is important for brain development, but we'll focus on some specific nutrients today. And we'll focus on their role in brain function and development, particularly on lipids, phospholipids, and sphingomyelin. And then we'll spend some time to understand what does the data show us? What is the clinical evidence for infants and children? And finally, as pediatricians, we have an incredible privilege, but it's also our responsibility to help every child achieve their full potential. And we'll end on that note towards the end of the webinar. So to start, let's focus on the anatomy or the structure of the brain. The numbers are impressive, with over 80 to 100 billion neurons, with a similar number of non-neuronal glial cells, and synapses which help the different neurons communicate with each other are over and about a quadrillion, with 100 kilometers of nerves and 600 kilometers of blood vessels. We know that the adult brain may be only 2% of the total body weight, but it consumes 20% of the total energy, emphasizing the importance of nutrition for the appropriate functioning and metabolism of the brain. So let's take a little bit of a deeper look into the fascinating organ. We know that while the brain is one complete structure, as we often refer to it, there are different parts of the brain which help with different functions. We know that the prefrontal cortex and the front part of the brain helps a lot with higher order functions, things like reasoning, judgment, problem solving, abstract thought. This prefrontal cortex drives a lot of the executive functions we all know are important in later life, as well as in childhood. Speech, hearing, recognizing faces, coordination, vision, somatosensory, and language listening as well as speaking are important centers in the brain which are critical in the developing child. But if we take a more microscopic look at the brain, we know that the brain tissue consists of the nerve cells, which we've all learned about in medical school, called neurons, but also some other cells like glial cells and oligodendrocytes, which really help not necessarily the structure, but also the function of the brain. We all know that myelin is an important component of the brain that wraps around these axons, and what they really help with is making the communication across these long and different lengths of nerves fast and efficient. We know from other literature that has been published over the years that as the brain develops, the myelin fraction increases and then plateaus. And this is just to show you MRI pictures of the developing brain, which shows the importance and increased presence of myelin in early life and as you grow up. But what's critical to understand is accompanying the structural development of the brain are different functions. While embryonically and anatomically, the brain from neuromodulation and neurulation starts as early as the first to second trimester of life, you know that once the baby is born, among the first functions that we can actually see the baby develop is seeing, recognizing faces, and then hearing. And then as a baby grows older, different functions have different levels of peaking. This is receptive language or understanding when they hear different sounds. And as they get older, from understanding words, they start to babble, and then comes speech production. And in the first later part of infancy, that's when higher cognitive functions develop, which continues to develop during the period of childhood, adolescence, and young adulthood. But what's critical during this period is the role of experience-dependent synaptogenesis. This is the more babies experience, the better their chance of achieving better neurogenesis in different parts of the brain, which is why interacting with the child is so important for the parents as well as other people in the household. And reading to a child and talking to a child is critically important in their early neurodevelopment to help them achieve their neurodevelopmental potential. As far back as the 1950s, we knew that as a child's brain grows, we see a more complex pattern of nerve formation in the brain. And Andrew Garner said, we see how early childhood experiences are so important to lifelong outcomes. How this early environment becomes embedded in the brain and changes its architecture was a fascination way back almost 70 years ago. Today, we understand this a lot better. In a publication from a few years ago, we now know that different parts of the brain develop at different times. Things like neurogenesis and neuronal migration start during the early fetal development. Astrogenesis, oligodendrocytes, myelination starts a little bit later, but continues throughout that critical period of childhood and into young adulthood. And the process of synaptic pruning, growing some areas, curbing some things which are not appropriate, continues throughout the period of childhood and young adulthood. So this slide brings to light the advances that have happened in our understanding of the structural neurodevelopment of the developing brain. Now that we know that the brain develops because of various structural and functional components of the brain, let's look to see what are critical factors that influence this development. We know that genetics has a large part to play with it. And the genetic expression of the genes that run in families is critical in helping a child develop their brain and their neurofunctional capacity. But there are environmental factors as well. Molecular cues guide the development and are dependent on the experiences that a child achieves in these early formative years, which is why interaction, engaging, reading, and talking is so critical. And then within the environmental factors, we know from published data that the socioeconomic status as well as the nutritional status is key. Other factors that really play a difference are social interactions, urbanization, pollution, social mobility, and stress. But let's look as to the data that convinces us on the role of nutrition in early brain development. Here are two charts that convince us that nutrition is important. From the World Bank publication back in 2018, this chart shows the blue shows the volume of the total gray and white matter in the brain of children who are not malnourished. And the yellow bar shows the volume of the total gray and white matter in children who are malnourished. And you can see that there's a statistically significant difference in the children who are not malnourished by the bars in the blue compared to the children who are malnourished, showing that nutrition is important. Another factor we talked about is socioeconomic status. And this shows the difference between high socioeconomic status, mid socioeconomic status, and low socioeconomic status on the volume of total gray matter in the developing brain across different periods of early childhood. And what this data basically convinces us about is that nutrition is important along with other factors in helping a child achieve their neurodevelopmental capacity. Why nutrition is so key among these other factors is because it's one of the modifiable factors. It's one of the things that you can educate and generate awareness about to help children get the nutrition that they need during this critical period. So what are the various brain development processes that we've seen that are supported by nutrition? These are myelination, structure and functional brain connectivity, the metabolism of the brain, and the neurotransmitter function. Let's look at nutrition in a little bit more detail. What is well known, universally accepted, and continues to be emphasized is the importance of breastfeeding and breast milk as the best way to feed a child when they are born. The WHO recommends exclusive breastfeeding for the first 6 months of life, and beyond that, continued breastfeeding to 2 years and beyond. We know that children who are exclusively breastfed have lower risk of certain respiratory and gastrointestinal conditions. We know they have less risk of certain conditions related to metabolic programming, be it overweight or diabetes. And there are differences in allergic and atopic diagnoses between children who are exclusively breastfed and children who are not. What we also do know is that optimal brain and cognitive development is significantly different in children who are breastfed, and desired as a great outcome for these children. So we take a look at this a little bit more to understand what are the specific breast milk components that provide these optimal brain and cognitive development in exclusively breastfed children. In a study that was done in the US, where they looked at about 62 exclusively breastfed children and 88 formula-fed children, they aimed to examine the longitudinal trajectories of brain and cognitive development in children who were exclusively breastfed as compared to children who were formula-fed for at least 3 months. And they were hoping to examine the development between children who received different kinds of nutrient components from the different formulas. What this study taught us, as you can see by the graph with the blue versus the yellow, that exclusively breastfed children had higher levels of myelin water fraction in the corpus callosum of the body of the brain compared to children who were not exclusively breastfed. The results of the study, which were done to really understand what is the difference in the nutrient composition, showed us that a retrospective analysis of the data taught us that certain nutrients showed a significant association with myelin content. These included nutrients like folic acid, DHA and ARA, phospholipids like sphingomyelin, phosphatidylcholine, and what we all well know is the importance of iron. And this observational data provides the correlation, but not necessarily conclusive, on the supplemental effects. So let's look at this data a little bit more. What they found was babies who were exclusively breastfed had the highest myelin water fraction, as you can see here by the blue bar. But different nutrient compositions, which we know exist in different formulas, had varying levels of myelin water fraction. The way they looked at some of the neurocognitive outcomes in the study was with a score called the Mullen score. And the Mullen score looks at ELC, which is an early learning composite score. What they looked to identify in this data was to see, okay, we know that nutrients contribute to myelination, but does myelination correlate with learning? And what you can see here through the red line is that there is a correlation between the myelination water fraction at 4 months of age and the early learning composite score. And when they looked at the specific nutrients which can impact myelination, now that we know that the correlation exists between the myelination water fraction at this particular age and the early learning composite, these nutrients were sphingomyelin, DHA-ARA, choline, a B vitamin like folic acid, and iron. What they also showed from this observational data was there was a difference depending on the different sphingomyelin content. There was a difference in the myelin water fraction between high sphingomyelin content as the dark black line and low sphingomyelin content as the gray line. What they also found was in different regions of the brain, there was a significant relationship between dietary sphingomyelin and myelin content at 12 and 24 months of age. So from this observational data, as well as multiple other human studies that have been done over the years, we now know that a link exists between myelination and cognition. And among the various factors that we consider when we think about cognition, these include general cognitive ability, language and reading, working memory, the speed of processing, and sensory reactivity. So let's take a look at one of the features that when we talked about the various factors that impact neurodevelopment and neurocognitive function, we know that nutrition plays a role. We know that myelination correlates with some of these cognitive outcomes. And we know that certain nutrients impact myelination. And what we're going to focus on today is on lipids, particularly phospholipids. When we look at phospholipids, we know there are different kinds. We know there are glycerophospholipids, things like phosphatidylethanolamine, phosphatidylcholine, phosphatidylinositol, and phosphatidylserine. And we know that there are sphingolipids, particularly among them is sphingomyelin. But we know that children who are exclusively breastfed have very optimal and desired outcomes when it comes to neurodevelopment and cognitive outcomes. So let's start by looking at the composition of these nutrients, particularly phospholipids, in human milk. What we know from the various studies on human milk analyses is that fat accounts for about 50% of the total energy, out of which is contributed about 0.2 to 2% by phospholipids. And a very general way to think about phospholipids is 5 predominant phospholipids. The 3 predominant ones, which account for anywhere from 60 to 80%, is phosphatidylethanolamine, phosphatidylcholine, and sphingomyelin. And the 2 less predominant phospholipids are phosphatidyllinositol and phosphatidylserine. And when you look at the body in general, and when you look at the brain more specifically, to look at what are the various components contributing to this, you will see that lipids are about 10 to 12%. And when we look more specifically at human milk, what we learn is that sphingomyelin, which is particularly rich in the myelin sheath of the central nervous system, and as we've seen from previous slides, is very important for the integrity, function, and for fast and efficient communication across the neurosystem of the brain, that both of these, sphingomyelin and phosphatidylcholine, are among the most abundant phospholipids in the human milk fat. Breastfeeding, as we've seen clearly from the various reviews across the years, provides the ideal form of nutrition for infants, and human milk is the only source of nutrients when it comes for the exclusively breastfed babies. But important to realize when we talk about sphingomyelin and phospholipids is that they can also be synthesized innately by the body, which is called as de novo synthesis. So let's look a little bit more at the various phospholipid components in human milk. What I'm showing you here by the red boxes is that, as you can see, that sphingomyelin comprises an important composition of total breast milk lipids across the time period at 30 days, 60 days, and 120 days. And you'll see that total phospholipids remain important across the lactation phase, be it 30, 60, or at 4 months of age, showing the importance of the compositional importance of these phospholipids in human milk. When you think about phospholipids, a lot of them are commonly called as polar lipids. And these polar lipids really comprise of a hydrophilic head and a hydrophobic tail. But what's important to realize about these polar lipids is that they form important structural components of various neural tissues. They have a role to play in neurogenesis. They have a role to play in synaptogenesis on how nerves communicate with each other, and a critical role to play in myelination, in the formation of myelin, but also in the communication. And the peak rate of accretion overlaps with critical neurodevelopmental milestones. So a good way to summarize it is to understand that these phospholipids are important for brain connectivity and for fast and efficient brain communications. And when you look at the different kinds of myelin lipids that we've talked about already, you will see that there are different ratios that you can find. But these myelin sheaths are very important in helping the communication across the different axons and the way the brain communicates across the central nervous system and the peripheral nervous system. So now that we know that phospholipids form a critical portion of the brain, let's look a little bit about the brain composition of phospholipids. And what I wanted to show you here, that both at 10 months of age and at 6 years of age, you can see the important lipid composition of the brain. When we look more specifically at these glycerophospholipids and these sphingolipids, we see that there's a growing importance of these glycerophospholipids across the time period of early childhood. Phosphatidylcholine and sphingomyelin particularly are very important and quantitatively differentiated from some of the minor phospholipids that are important in brain composition. So to summarize a lot of the anatomic and structural data on the strong correlation that exists between nutrition, myelination, we know that phospholipids are major components of the brain. They're important for integrity, they're important for metabolic pathways, but most important, the fast and efficient communication across the brain. This shows that sphingomyelin, phosphatidylcholine, serine, and ethanolamine accounts for up to a third of the dry weight of these myelin sheaths. But let's look and spend the last few minutes looking at the clinical evidence. This study, published in Japan in 2013, was a small study that really looked at preterm babies. We all know the neurocognitive outcomes associated with prematurity. And this study was among the first studies to show that giving babies these specific components of phospholipids may make a difference in some of their neurocognitive outcomes. So the objective was to examine the effects of sphingomyelin on mental, motor, and behavioral development of preterm infants. In this randomized, double-blind, control trial, they took 24 very low birth weight babies, and they actually gave one group a formula that contained up to 13% of sphingomyelin versus the other, which contained a lower level of sphingomyelin. What they aimed to show in the study was that giving babies who were breastfed in this a supplemental nutrition with a formula that contained 20% of sphingomyelin versus a low level of sphingomyelin impact their developmental outcomes. What you can see here from the results of the study is that there was a statistically significant difference in the treatment group that were breastfed but got a formula as supplementation that contained higher levels of sphingomyelin had significantly improved outcomes on various neurobehavioral scores, particularly around behavior rating scales, around the Fagan test, and around sustained attention. So let's look a little bit more at what these neurocognitive various testing tells us. We're all familiar with the Bayley score, which helps to evaluate mental and psychomotor function. The Fagan test really helps to measure the novelty rate preference as an indicator of intellectual development. They looked at visual evoked potential test, which tests the level of myelination and neurotransmission speed via the optic nerves. They looked at the sustained attention test and memory test, which was significantly improved in the test group, which also are indicators of neural development. What did they show in the study? That the percentage of sphingomyelin in total phospholipids in red blood cells was significantly higher in the sphingomyelin fortified group compared to the control. They showed that behavior, aspects of cognition and visual test results at 18 months was significantly better in this 8-week study in the sphingomyelin fortified group than compared to control. And they concluded that nutritional intervention with sphingomyelin fortified milk has a positive association with neurobehavioral development of these infants. So that was one study in preterm babies. Let's look at another study, which was a larger study that included about 160 children divided into 2 groups. There were 80 children who were formula fed with an experimental formula that contained a protein-rich lipid membrane fraction and a standard formula. What they found was that at 6 months of age was their primary outcome of intervention. But Baileys were looked at 12 months of age. And they found at 12 months of age that the experimental formula group received higher scores in the cognitive domain of the Bailey scale than in the standard formula group. They concluded that nutritional interventions were critical. And in my last slide, I just want to talk about the role of pediatricians and our responsibility. First, I want to thank all of you for your commitment and dedication to improving the lives of children everywhere, particularly in India. But early childhood provides all of us as pediatricians the ability to see children frequently, more so than at any other time period during their life. So while we have an incredible privilege, it's also our responsibility to help educate parents and caregivers on the role of nutrition in early childhood, that breastfeeding remains the best way to feed a child. But consider the importance of nutrients and nutrition when making feeding decisions and educating parents and caregivers. Thank you. So I see that there are a few questions come in, and the first question is how phospholipids can help in brain development. So as I've shown you the data, the way we look at phospholipids is that phospholipids is a group that contains both glycerophospholipids as well as sphingolipids. And these are important components of various tissues in the body that tend to have significant levels in certain structures of the brain, particularly when we look at the lipid composition of the brain, but also the myelin sheaths. And what we believe these phospholipids do is really contribute to the structure and the formation of myelin, which helps in the fast and efficient transmission of various communications between different parts of the brain. Another question that comes in is, what is the role of different vitamins and minerals in brain development? What I can share with you is from the data that we've seen from the cohort that was down at Brown University, we clearly saw that there are certain nutrients which really stand out when it comes to brain development. Iron, which is a mineral, is one of those critical nutrients which are very important. Certain vitamins, particularly the B vitamin folic acid, also stands out as something that's critical, which we know also has a big role to play in the prenatal period in reducing the risk of conditions like neural tube defects and spina bifida. While we've known about the role of DHA and ARA, what I was showing you a lot of today is the role of these lipids. We've always known that they are important both in brain development, but now we know that specific lipids, particularly phospholipids, including sphingomyelin, now have a critical role to play in brain development. Another question that has come in is, what is the best way to increase cognitive potential in your child? This is a fantastic question. Well, I've shown you all the factors that are associated with normal brain development and function. These are genetics, environmental factors, social factors, socioeconomic status. But then there are some factors which are modifiable, nutrition being one of them. And I've talked a lot about the role of nutrition, but also social engagement and social interaction. So I just want to say that social engagement, interacting with your child while you're feeding your child, whether at the family table later on, or while breastfeeding a child early in life, is very critical to have that interaction, to talk to your child, to read to your child, to sing to your child. And then as that child grows older, that child interacting with other children as well is critical for that experience-dependent neurogenesis and synaptogenesis. And these are key ways to help your child achieve their cognitive potential. The last question that I can take is, what is the importance of genes? So genetics plays an important role, and it is critically important in defining the various potentials that a child may achieve. However, the important things to realize about genetics, it is not what we consider an easily modifiable factor. So other factors which might allow themselves to be more modifiable are things like the environment in certain cases, social interactions in a lot of cases, and nutrition. So genetics are very important, and they play a critical role in the child achieving various structural and functional neurodevelopment and outcomes, but it is less amenable to modifying than other factors like social interactions, experiences that you give the child, and nutrition. So I want to end by saying thank you to everyone for your time, for your commitment to further all our understanding on early childhood nutrition, and finally to thank you for your incredible role as pediatricians that ensure that we raise together with parents and caregivers the next generation of children that achieve their full potential. So thank you very much, and have a good rest of the day.